My name is Nick Elder. I'm a third-year graduate student at the University of California, San Francisco in the Developmental and Stem Cell Biology program. Today, I'll be sharing with you some of the graduate work I'm doing in the McDevitt lab. So I'm interested in spinal cord development. And more specifically, I'm interested in a population of ventral neurons in the spinal cord called V2A neurons. These are excitatory interneurons that are located throughout the spinal cord and which make direct connections onto motor neurons. I'm also interested in this population for its therapeutic potential in treating spinal cord injury. So a key aspect of V2A neurons is that they express a transcription factor, CHX10. However, recent studies have begun to identify diversity within the V2A lineage. For instance, as V2A neurons mature, they resolve into two further subtypes, type 1 and type 2. One of these types maintains the transcription factor CHX10, whereas the other subtype downregulates the CHX10 expression. And this isn't a purely stochastic process either. These two populations are differentially distributed along the spinal cord. And so there are indications that this is a regulated process. There's also some studies that show that there are differences in their axonal projections. And so the objective of my research is to study the regulatory and developmental mechanisms that lead to V2A diversity along the rostral-caudal axis, and to determine the impacts of V2A diversity on neuron function. The long-term goal of my research is to gain a better understanding of spinal cord processing, as well as to better leverage the V2A neuron populations for therapeutic uses. And so in my research, I'm trying to generate human V2A neurons to study in vitro. And so in order to do this, I'm starting with a population of human stem cells. I'm then transitioning them into an axial progenitor state. These cells are termed NMPs. And this is a crucial process in this differentiation because in the NMP state, Hox genes are induced, and these confer axial identity to the downstream neurons. These axial progenitors are further differentiated to spinal neurons, where I can study the V2A population. To further facilitate my research into the V2A neurons, I have engineered a V2A lineage-traced cell line that marks all of the V2A neurons with GFP expression. This project is still in the early stages, but I look forward to sharing more with all of you at my poster. Thank you.